Hi everyone, still with BIO511 Genetics, and we will now move on to Euphenics. So what is Euphenics? It's actually a new term to differentiate from eugenics because eugenics is associated with more negative than positive things. So Euphenics is a new term they use. So it literally means good appearance or making phenotypic improvements. So phenotype is something that you can see, like the color of your hair, the color of your eyes, um, and everything that you can see on somebody or on a plant or anything. So euphonic is we want to turn something into into something good. So it refers to medical and or genetic intervention designed to reduce the impact of defective genotypes on individual. For example, the use of insulin by diabetics and the dietary control of newborn phenylketonuria. Okay, so children born with the genetic disease phenylketonuria (PKU) can avoid the expression of their disease by staying on a low phenylalanine diet and avoiding major sources of phenylalanine, such as diet soft drinks sweetened with aspartame. So there is a thing which is trade name NutraSweet. Now we move on to genetic advances in agriculture. So selective breeding and hybridization technique has had the most significant impact in agriculture. Plants have been improved in four major ways. One is to enhance the potential for more vigorous growth and increased yields, such as crops as barley, beans, corn, oats, rice, rye, and wheat. So we have enhanced the potential for these plants to be more vigorous in growth and to increase more yields and to increase its resistance to natural predators and pests. Also, production of hybrids exhibiting a combination of superior traits derived from two different strains or even by two different species, such as the triticale, which is a hybrid grain derived from wheat and rye, produced as a result of applied genetic research breeding experiment. And then number 4, selection of genetic variants with desirable qualities such as increased protein values, increased content of limiting amino acids, which are essential in human diet, or reducing vulnerability to adverse weather conditions because some plants cannot live in harsh weather. So we want to enhance this feature. So we hybrid it with other plants that can withstand the weather. So this is an example of the a hybrid plant, the triticale. So it is from wheat and rye, and it produces triticale with a combination of these two. The grain of wheat, rye, and triticale. So triticale grain is significantly larger than that of wheat. So it is created by a Swedish researcher in 1875. And it has more gluten. So this triticale has more gluten than rye and more protein than either wheat and rye. So the combination of these two produced a plant that has more gluten and more protein than the original plant. And then there's also this another example, which is a high-yield semi-dwarf variety of rice called the IR8. A part of the second green revolution was produced by crossbreeding two parent strains of rice, Pita from Indonesia and DGWG from China. So the shorter stalks of the new variety allow the plants to support the larger head of grain without toppling over. So usually we produce hybrid so that it can have better features, combining the good features of both the parent species. And we also have development of superior breeds of livestock. Selective breeding has produced chickens that grow faster, yield more high quality meat per chicken, and lay greater number of larger eggs, so that we can sell it more. We can have more values for the chicken can. And in larger animals, including pigs and cows, the use of artificial insemination has been particularly important. So this is one example. The American boer goat is one of the hardiest of all livestock breeds. Hardiest means it can live in harsh conditions. They're tough lah. It's tough. Bred for dominance, the breed distinguishes itself by carrying superior genetic characteristics for goat ranching. Boer goats have a high resistance to disease and adapt well to various climates and terrain. Internationally and now in the United States, the boer has been used to improve other breeds through aggressive cross-breeding program. So this is the superior breed and used to improve other breeds. And then we also have this, the Brahman and Angus that produce Brangus. So the combination results in a breed which unites the traits of two highly successful parent breeds. So the Brahman, through rigorous natural selection, developed disease resistance and overall hardiness, while Angus are known for their superior carcass quality and also extremely functional females which excel in milking ability and outstanding maternal instincts. So Brangus has both the qualities of Brahman and Angus. And then we also have genetic advances in medicine. For example, we have genetic counseling, which provides parents with objective information upon which they can base rational decisions about parenting. And we also have immunogenetics, which has made possible compatible blood transfusion as well as organ transplants. We also have histocompatibility tests, which is the discovery of genetically determined tissue-bound antigens. And this histocompatibility is a property of having the same or mostly the same alleles of a set of genes called the major histocompatibility complex. These genes are expressed in most tissues as antigen, and the immune system makes antibodies. And we also have the recombinant DNA technology, which creates a new bridge between DNA molecules or segment of DNA molecules. So this is the process of cloning DNA in a plasmid, in which our gene of interest, the foreign DNA, is inserted into a plasmid vector which carries the antibiotic resistance gene, and then it will form a recombinant DNA molecule, in which this molecule will be introduced into the host cell. And then we select the cells containing recombinant DNA molecules by growth in the presence of antibiotic. So those cells that live is the one that carries our gene of interest, because the plasmid has antibiotic resistance gene. So that's how we clone DNA in a plasmid. And then there's this another process of using the restriction enzyme. For example, this is the EcoR1, which produce fragments with sticky ends and then mix these fragments from both digestion. So both parent DNA was digested by the same restriction enzyme. And then it is mixed in the fragments are mixed together so that they can base pair and incubate with DNA ligase to link both strands and make a recombinant DNA molecule. Okay, so in medicine, among others, we have insulin, which is a hormone required to properly process sugars and fats to treat diabetes, and now it can easily produced by bacteria. So for those diabetic patients that cannot produce insulin by themselves, they can now have insulin to be injected in their body, produced by bacteria. And for people with growth hormone deficiency, there is also a hormone that is now easily produced by bacteria. So that's it on Euphenics, which basically means we combined the good characteristics of two different species or strains to create a better one. So what do you think of Euphenics? And say if you know that you're eating a recombinant breed, will you continue to consume it? Will you feel safe to eat recombinant cereal or something? Because maybe you don't know. When you don't know, you just eat it, right? But if you know, how would you feel about it? Then that's something that we will discuss in the ethics section. Okay?